Hey everybody, how you doing? We are starting our third quarter here finally, and we're the third quarter can is mostly made up of genetics, like pretty much the entire quarter in one way or another. You can relate it to genetics. So we're going to start with the cell cycle, which is like mitosis and meiosis, along with a few other things. Um, and then we'll get into other aspects of genetics down the road. But for now, first unit's going to be on the cell cycle. So we're going to talk about mitosis first. Um, so big questions: why do cells need to divide, and how does this allow for the continuity of life for individuals and species? So you begin as a single cell, right? And that one becomes two, and the two becomes four, so on and so forth. And off we go, and we keep dividing and dividing and dividing. And pretty soon that group of cells turns into like a hollow ball of cells, which is called a blastula. And then after that, the hollow ball of cells actually gets an indent, and it starts pushing into the middle. And that is called the gastrula, which ultimately becomes your anus. So, fun fact. And it keeps going and going. And after so many days, so many weeks, we end up uh, with a full fledged newborn, right? Because these cells keep going and going. So, why divide? The simple answer is to preserve the continuity of life, right? In order to survive, an individual has to replace damaged cells. And in order to grow, cell production must be greater than cell death. Okay, so that's really the main few reasons why you have to go through this process of mitosis. So a couple pictures here. There's a dividing amoeba right there, dividing bone marrow cells, dividing sea urchin embryos. So lots of different types of cells are going through mitosis. For a species to survive, individuals must be replaced, right? So reproduction rate greater than death rate, and that kind of goes along with cells, right? If you're going to grow and if you're going to survive, then your new cell rate has to be greater than your cells that are dying. So here is the cell cycle. Okay, so lots of different phases here as we go around, and we'll get into these more and more as we go. But you can start with G1, and you'll notice G1 takes up a pretty good portion of that pie, right? Of that pie chart. And that is where most of the cell growth happens, as well as just that's a cell being a normal cell. Okay, G1 cells are doing normal cell things. In S phase, the red part there, that's where DNA replication is happening. That's where you're copying the DNA so that you can have a set of DNA for the old cell and a set of DNA for the new cell. And then in G2, you're preparing for that division. And then that M stands for mitosis. And there's actually four phases within that M that you might remember, and we'll get to in a second. But there's lots of happening within that M. And then we have something called G0, which you probably didn't talk about a lot in regular bio. And it's a non-dividing stage. And this is most cells most of the time after they get to their kind of post-adult years, kind of retirement. G0 is kind of retirement for a cell. They live out their lives doing that. There's a name for this as well, which we'll get to in another set of notes. Also want to draw your attention to interphase. Interphase is G1S and G2 collectively. Okay, so all of this. So if you look at G1, S, and G2, all of that area, then that's what this orange is on the outside. That I that I just passed right there is interphase. So interphase takes up a massive amount of time compared to mitosis, right? So most of the cell, a lot of time, the cell is spending in some version of interphase, whether it's G1S or G2. So in order to go through this, we kind of have to look at the anatomy of DNA and the anatomy of a chromosome again. So DNA, you guys know, starts with the helix, right? And then what happens is DNA gets coiled around these proteins that are called histones. And all of those bluish purple little dots right there, or little spheres in there, those are the histones, okay? And as they wrap around, the DNA wraps around those histones, then we say the DNA and the histone, all of that together is a nucleosome, okay? So Histones are just the proteins. A nucleosome is DNA and protein, and it kind of looks like beads on a string. That's that's kind of the general term, what we call when it starts wrapping around them. It looks like beads on a string. So DNA and nucleosomes then coil up together really tightly, and then they keep going, and that's what we call chromatin. Okay, and chromatin is still very loose DNA. That is not really tightly coiled. It's a loose coil. Okay. And so chromatin keeps going, and ultimately chromatin will coil up really tightly into a chromosome, and it will condense and coil into a chromosome. Okay, so most eukaryotic cells have two copies of every chromosome, and then they form identical pairs, right? Because they're copied. So what this picture is, you see where they look like an X, kind of like where I have my fingers together. That's usually one chromosome that then was copied, and now you have two. And my fingers are different lengths, but you get the idea that these should be identical because it was copied from one or the other. Okay, so um, this, just to kind of get into some semantics here between chromatids and chromosomes. So you have a chromosome and you copy it. This is still a chromosome, okay? But each piece, each one of these fingers is a chromatid, okay? A sister chromatid. Okay? So we'll get to some more of that in a minute, but that's kind of how you deal with the terminology, okay? And where they get attached. And honestly, it's more like this. It's more like chromosomes are more like this. I know they show you X's in textbooks a lot. They show you X's like this, but really they're like this and they just touch in the middle. Okay. And where they touch, that little spot right there is called the centromere. Okay. So remember the difference between prokaryotic and eukaryotic chromosomes. The one I just showed you would be eukaryotic, and they are more linear. Remember that a prokaryote has a circular chromosome. One circular chromosome is all prokaryotes have. Eukaryotes have many linear chromosomes. Okay, and you can see as far as cell division in a prokaryote, and we'll mention this again a little bit later as well. But you have your basically have your circle of DNA, and then it copies through replication. So now we have two circles of DNA, and then the cell is going to split, and that's pretty much it. It's super simple. Okay, it's going to be a little different for eukaryotic division. So here again is a slide about chromosomes versus chromatids. So you have a chromosome, and when you copy them like that, you have sister chromatids are each one of the fingers in that. These are each chromatids, right? Together, it's still a chromosome. Okay, the whole thing is a chromosome still made up of two sister chromatids. Okay, and then when they split again, this is a chromosome and this is a chromosome. And we don't really talk about chromatids unless they're together. Okay, so hopefully that helps you sort out some of the terminology. More terminology, haploid versus diploid. And I remember a lot of regular bio students struggle with this concept. So hopefully it'll make a little more sense the second time around here. Haploid is just one copy of every chromosome, and diploid is two. Okay, so we use the letter N and 2N for a reason. N is just the number of chromosomes, whatever number it might be for that particular organism. Because all the numbers I'm going to show you have to deal with humans, but we don't have the same number of chromosomes as everything else. So humans have 23 pairs of chromosomes or 46 pieces of DNA. Okay, so 23 pairs. So we have 22 chromosomes that are numbered 1 through 22, and then we have our X and Y, our sex chromosomes. We have 23 pairs of them, or 46 pieces of DNA. Okay, so 23 would be like our haploid number, and 46 would be our diploid number. Okay, so how many pieces of DNA would be present in G1 and then G2? So maybe pause the video, stop and think about that for a second, um, and see what you come up with. So for G1, okay, G1 is happening. That's a cell doing normal cell things, right? The cell would have 46 pieces of DNA. Okay, it didn't say chromosomes, it said how many pieces of DNA. So G1, you would have 46 pieces of DNA. In G2, how many would you have? Well, what happened between G1 and G2? What actually happened? That S phase happened, that synthesis phase, where we copy the DNA and we replicate the DNA. So if G1 has 46, G2 should have 46 times 2 because we copied it. So it should have 92. Okay. So G1 would be 46, G2 would be 92. Okay. This is just kind of an interesting table here of the number of chromosomes that a lot of different things have. Um, because, like I told you, we have 46 pieces of DNA. Your cat has 38, okay? But your dog has 78. Your dog has 78 chromosomes. How crazy is that? There's also some other weird comparisons in here to humans. If you look at, let's say, a housefly only has 12. That probably makes sense to you, right? It has a lot less. But what about a potato? A potato has 48. Has two more than we do. Okay, it's kind of weird, right? So look up and down this group of things here, and it's kind of interesting to see how many chromosomes different animals or plants and fungi have compared to us. That's why we use the N and 2N system, the haploid and diploid, because we're not always talking about the same number. It's not always 46 like it is for us. All right, running through the phases themselves. Okay, so interphase, growth, replication, and preparation for division, right? Those first three bullets are basically G1, S, and G2. Okay, most of a cell's life is spent in interphase. Cell looks normal, doing normal cell things. And just for future reference, when we start talking about centrioles over here in the red circle, centrioles are only in animal cells. Plant cells will not have any centrioles. Okay, so keep that in mind. So in prophase, so now we're talking about the first phase of mitosis, right? Interphase is G1S and G2. Prophase is the first phase of mitosis, okay? So in this case, chromosomes condense. That's the big thing that goes on. They go from chromatin, which is unwound, spaghetti-like DNA that's all tangled in the nucleus, and now it starts condensing and they get really short and they get to look like those X's that you usually see. The nuclear envelope breaks down because we're going to have to do some copying and make another cell, so we got to break down the nucleus. The mitotic spindle begins to form, and centrioles divide only in an animal cell. Okay, but the main things you can see, actually, let me go back one. So if you look at interphase, you can't really see anything distinct inside these cells. They're all just a big ball of spaghetti. There's nothing really distinct about them. But if you look at prophase now, which is the next phase, you definitely see these are much thicker and shorter little pieces of DNA in there. And you can see them because of the stain that they use on the microscope slide. You can actually see them in there much better, and they're condensed, which means they're darker. And you should be able to tell prophase from interphase that way. ProMeta wasn't even around when I was a student. They've kind of invented this one. I think they thought there was just too big a gap between prophase and metaphase, so they came up with prometaphase. And this is where chromosomes begin to migrate to the equator, and then the spindle is completely formed. The spindle is going to be this group of like proteins that act as like the strings or the connectors that are going to grab the DNA and pull it to each end of the cell. So that's the spindle, which we'll focus on here in a minute. But yeah, ProMeta is just getting things organized. You can see the spindle fibers over here have now attached to the centromeres, right? They've grabbed these pieces of DNA and they're starting to jockey them into position. Okay, they're trying to get them all the way to the equator, to the middle, so then they can be separated equally, which is what metaphase is, right? Meta middle. That's how you always remember metaphase, when they are lined up at the middle. Okay, so again, here they are lined up, the DNA is lined up at the equator of the cell. You know this is the DNA because it's the darker stained stuff. Okay, so all that right through there, and it's all lined up at the middle. If we go to our cartoon over here, all the centromeres are pretty much lined up along that line right there, right along the middle of the cell. And this allows the spindle fibers to attach to all of them and then hopefully pull them equally toward the different poles of the cell. Okay, and then these little yellow guys right here are supposed to be the centrioles, which are kind of the anchors that the spindle fibers are attached to, and it gives them something to like pull against to separate the DNA. Okay, And it says right here the spindle fiber is attached to kinetochore proteins in the centromere. I'm going to show you that picture here in a second. So here are the kinetochores. Okay. So kinetochore proteins are like these, what looks like a blue disc right in here, okay? So those are the kinetochore proteins at the centromere. The microtubules, which is part of the spindle fiber. Remember when we did microtubules back in cytoskeleton days, and we said they help in mitosis and meiosis? So this is where that knowledge comes back to help. So you have microtubules that form the spindle fibers, and they're attaching to the kinetochore proteins, and they're going to be what moves the chromosomes around and ultimately separates them, okay? So the centrioles at the top end, um, spindle fibers, and then what happens is the microtubules do not really work like bungee cords. Okay, they're not stretchy. Okay, they either lengthen or shorten by the addition or subtraction of what's that protein that makes up microtubules that you learned in first semester? Should be tubulin, right? So remember that video I showed you where the tubulin gets added or subtracted very quickly? So that's what's happening here. So when this um, spindle fiber is pulling the chromosome toward this direction, that means all of these microtubules right here are getting shorter, and it means all these microtubules are getting longer. Okay. So because they're like little pieces of Legos, they get added really quickly to lengthen things or then shorten things. You take them away. Okay. So now they're migrating to the poles of the cell. The chromatids are split at the centromere, and we say sisters separate, right? Because these are sister chromatids that are now separated, and they are migrating toward the two poles of the cell. And this is how we know that they aren't bungee cords. This is a little experiment. If you look right here, so see this gray section right here and right here. Okay, so they put a little marker on there that attached to the protein, and they could track whether it was moving toward the centriole or whether it was staying put. Well, if you look up here, here's our DNA, right? If you look at it, when the DNA is getting pulled this way, does the gray section of the spindle fiber move? Well, it doesn't. Look, it's all staying right there. But the DNA is definitely getting closer to the pole than it was here and than it was here, but this part hasn't moved. So they know that what happens is these little pieces of tubulin are just getting chewed up and they're getting released, and it's just making this microtubule shorter as it goes, right? And here's the kinetochore protein right here, and this is your chromosome. The big purple section is your chromosome, and oops, you have what's called a motor protein, which is the red part right here that's having it's kind of helping walk this guy back down this way, and then the tubulin subunits are coming off of it. Okay, so that's just kind of how they figured out that the microtubules actually shorten versus like stretch. Okay, telophase, the last phase of mitosis. Chromosomes decondense. So now they are going from little tiny chromosomes back into chromatin, and they're not as coiled. Okay, so they're stretching out. Nuclear envelope is going to reform because we want to protect the DNA, right? So this nucleus is forming and this nucleus is forming around over here, and then we're going to have cytokinesis, which is going to split it for us. Cytokinesis is not a part of mitosis. It is right after mitosis, okay? So mitosis is prophase, metaphase, anaphase, telophase. Cytokinesis happens after it, and this is just the splitting of the cytoplasm, the cutting of the cytoplasm. And it happens differently in animal cells and plant cells, okay? In an animal cell, there's a contractile ring of a microfilament. Remember, we talked about this as well when we did cytoskeleton. So there is a lasso. Okay, there's kind of like a lasso that goes around it, and it works like a zip tie, where when you feed a zip tie in, and then you keep pulling the string on it, and it keeps cinching and cinching and cinching and cinching, and so it keeps going down on that cytoplasm until the two cells pop apart like that. So you get to my little picture. So pretty soon that microfilament contracts so much that it's going to split the cytoplasm there. Um, and remember what protein makes up the microfilament? You guys remember that one? Should be actin. Okay, actin is the protein that makes up the microfilament, which is doing that constricting. For a plant, it has to work a little different. You can't just throw a lasso of a microfilament around a plant cell because why? Because it has a cell wall, and that cell wall is way too strong. So you can't just cut a cell wall like you can a cell membrane. So in this case, it has to get built from the inside. Okay, so if you look here, there's vesicles dropping off material here in the middle, and before it's totally connected, we call this a cell plate. Okay, so the cell plate is forming, okay, right down the middle. Once it gets totally connected all the way across, then it becomes a cell wall. Okay, but until then, it's a cell plate. And these vesicles, what do you think they're dropping off right here? What makes up a cell wall? Anybody remember the polysaccharide that makes up a cell wall in a plant cell? It should be cellulose. Okay, so vesicles are coming along and they're dropping cellulose right here to make that cell plate. And ultimately, once it's all the way across, that is called a cell wall. And then you get your daughter cells, okay, which is kind of the result of mitosis. Okay. So this is kind of a fun game here. In class, I usually call out a phase and tell them to find them for me. So if I were you guys, I would pause this and see if you can you find interphase? Can you find prophase? Can you find metaphase? Can you find anaphase? And can you find telophase? Okay. I would work through these and see if you can tell which ones are which. Okay. We're actually going to do a lab in class, which is it's going to become useful if you can tell which ones are which. So take that a shot. I'm not actually going to give you the answers right now. See if you can go through and label a few of these and go back and look at the diagrams if you need to. Okay. And lastly, just kind of wanted to touch on the evolution of mitosis. So bacteria, we've talked about a little bit, right, with binary fission. Okay, so and how the two loops just kind of, or the one loop copies into two, and then the cell divides, right? And this is binary fission in bacteria, also called rolling circle replication, by the way. There's kind of another name for it. It's actually pretty similar to how a lot of eukaryotic cell division goes. May not seem that way, but there are a lot of the same proteins that do the replication and process over here, and it does the same process down here to where you get into these eukaryotes and these more advanced forms of mitosis. A lot of the proteins are the same between the prokaryotes and eukaryotes, which lends itself to some sort of evolution, right? That it probably just evolved, the process evolved, and there's like some intermediate modes of cell division, like in these guys, these dinoflagellates and these diatoms and in some yeast. They're a little bit more complicated than the bacteria. They're not as complicated as the eukaryotes down here, but they do share some of the same proteins, like kinetochore proteins are in there. And so there are these intermediate modes of mitosis. You may not have thought about that before, but there is kind of an evolution of mitosis. Okay. Well, that should be a pretty good intro to mitosis for you. So go back through it if you need to. Here's some questions to help you out, and hopefully you can use that. Take care.